All right, welcome back to part 4 of transcription. By the end of this lesson, I'm hoping that you will be able to list the different types of RNA modifications and explain overall what the advantage of RNA splicing is. So, back in the early days of RNA research, and we were looking at bacterial genes, what we would realize is that the sequence in the DNA corresponds directly to the sequence in the messenger RNA, and the sequence of those codons in that messenger RNA correlates directly with the sequence of amino acids in that polypeptide. So this entire concept was collinearity. So when you see the DNA, the messenger RNA will be the same, and how the protein is laid out will be identical. When we started progressing past bacteria into the world of eukaryotic cells, one of the first things we realized is that the messenger RNAs aren't always collinear with their genes, meaning that there's a difference. And instead, we have coding sequences, which you now know are called exons, are interrupted by these intervening sequences, or what we now call introns. So transcription produces a pre-messenger RNA that corresponds to the entire genetic sequence. Those introns are later removed, or we use the word excised, and the exons that are left are reconnected together or spliced. So this entire process we've referred to as RNA splicing. Now, RNA splicing is only one of the modifications that RNA can go through, but let's start there and just think about it from how it was discovered and what it can do for a cell. And then later on, we'll get into the mechanics of how that splicing actually works. One of the first eukaryotic genes that was worked on in regards of messenger RNA is the gene for beta globin. And this is part of the complex that carries oxygen in our red blood cells. Cells that make this make tons of it, so it was really easy to work with. We could have endless supplies of messenger RNA for this. When we take the messenger RNA from this and try and hybridize it back to the DNA, we notice that something weird was happening. So here's an experiment where we take a region of DNA and we take a mature messenger RNA. We dissociate the two strands, or denature the double helix. So we have two independent strands. We allow this messenger RNA to base pair with the DNA that it is complementary to, or to the template strand. And what we would end up with is this type of structure here. And this is what happens in bacterial systems. So we would have a region of the genome that is double helix. Then all of a sudden we'd get this weird bubble, and that bubble corresponds to where the double helix is blocked by the binding of this messenger RNA to its template strand. And then past that, you would have more double helix. So we'd refer to this as the R loop, or the RNA displacement loop. What happened when we took this into eukaryotic cells is, and we'll discuss it from the perspective of you already know what exons and introns are. So here is a piece of DNA. It has some exons and then an intron and another exon. It would make a transcript. This is what the transcript would look like. This is the pre-messenger RNA. It has the intron included in it. Afterwards, this is spliced and the intron is removed, and now you have this mature messenger RNA. If you were to take this mature messenger RNA and attempt to do this hybridization experiment, this is what you end up with. So you'd get a chunk of DNA that has a hybridized messenger to it, and then the whole region that corresponds to the intron would just be looped out. Okay, so this is just looking at if the binding of the template and the mature messenger RNA. Now, if you then take the coding strand and allow it to hybridize back on, what you'll notice is that the intron section is present there, and it can hybridize here. Over at the ends, you will get double helix and hybridization forming. But then here we will have these two R loops corresponding to where the two exons are. So the fact that this is what we got and this isn't what we got vexed us for quite a period of time. It took us a while to work out the details of why this was happening. But this type of structure is easily visible under electron micrographs. So here's an example of that. So here's a diagram of what we're looking at here. So here we have an R loop, we have the intron loop, another R loop, and then the double helix DNA on both sides. So it's something that's very visible. Now, you might start to question, well, what is the advantage of putting these introns into our genes? Does it make it more complicated? It does, but it so happens that it's complicated in a good way. What this does is it leads to the possibility of what we refer to as alternative splicing. So a single pre-messenger RNA that has multiple introns might be spliced in several different ways. This will correspond to mature messenger RNAs that have different combinations of exons. This essentially gives us different forms of a gene based upon the gene only having one version of the gene. So here we get multiple proteins out of one gene. That is certainly more efficient if you are dealing with an ever-increasing genome size, which is what eukaryotes were dealing with. So the fact that you can get two or in fact more polypeptides out of a single gene is good conservation of energy and keeping the genome as small as possible. We will talk in detail about how the mechanism of splicing works, but let's take a look at an example here. So here we have, and this is just a hypothetical example. So here we have the DNA corresponding to exon 1, exon 2, exon 3, exon 4, exon 5. This would create a pre-messenger RNA that would contain all of those elements. And then you go through the process of splicing it together. Well, here we have different versions of that splicing action, right? So here we have a messenger RNA that has all of the exons, 1, 2, 3, 4, 5. Here we have a version that is missing exon number 3. And here we have a version that is missing exon number 4. Now, if you think about this in terms of how this might appear structurally, this 3 alpha helix domain here is corresponding to exon 1. This 2, this kind of helix turn helix motif is what exon 2 is. Exon 3 is this beta sheet loop and this little extra loop. 4 is this second beta sheet type loop, and number 5 is this carboxyl terminus alpha helix. So you can imagine that something like this loop might have a particular function in the protein, and this loop might have its own particular function that is different from the function of the first loop. So here we've set up a possibility that we can have a version of the gene that has these first 2 elements and the last element, which provide kind of a structural background, but then we have different functionalities built in by either having both exon 3 or 4, or only exon 4 or only exon 3. You can imagine that if this was involved in like DNA binding or something, that you could have one that, so let's say that this is involved in DNA binding, exon 3 is involved in DNA binding, exon 4 is involved in recruiting a protein. If we were to produce this, we would bind up the protein but not link it to the DNA. So this protein here might have the exact opposite effect of what protein A does. Similarly, protein C can bind the DNA but not bind the accessory protein, the crosslink. So you can see that having different elements and being able to mix and match, you can produce what could be oppositely functioning proteins out of a single gene. So here's a real example. This is alpha-tropomyosin gene. This is involved in muscle contraction. So this is actually a switch type of protein. It turns on contraction or blocks contraction. And here we have a bunch of different exons, and it has multiple different introns. And here are different versions of that final messenger RNA that we see in different tissue. So we will see in striated muscle, which is skeletal muscle, that we get one version of it. In smooth muscle, which is what we see surrounding like our gut, and it works more automatically, it has a different form. Here's fibroblasts, which make connective tissue. It takes on a different form, and in the brain it takes on a completely different form. So this allows different cells to specialize what version of this it is producing. And finally, here is a comprehensive list of all of the different types of RNA modification. So one is just simply simple processing. So we have a longer messenger longer RNA, and it has extra pieces to it. Those pieces are cleaved off and degraded, and then we get the final version. This type of process happens in ribosomal RNAs, where we can actually get multiple RNAs out of one gene. Here we have an example of splicing. Here is a modification called capping. So in our messenger RNAs, we add on a cap to the 5' end that acts at a specific function. We can also add on a poly A tail, and we'll talk about that later on. Sometimes we can actually directly modify, changing what the actual nucleotides are.